Hello everybody, welcome to my channel. Today I want to talk about the synthesis of T4 and also T3, thyroxine and also triiodothyronine, and also talking about a little about the follicular cell and what is happening inside them and how they can produce T4 and also T3 into the blood and secrete it into the blood. So as you see in here, this is hypothalamus and this is anterior and also posterior pituitary gland. They have relation with each other. I mean that anterior pituitary gland through the hypophyseal portal system has relation to the hypothalamus, and the posterior pituitary gland has relation because of the infundibulum. But here I'm going to concentrate on the anterior pituitary gland. And what? Here I have some nuclei which are called the paraventricular nuclei. These nuclei will synthesize TRH. It's a hormone. It will be released into the capillaries due to the net plexus, to the primary net capillaries of the hypophyseal portal system. And in here I have the hypophyseal portal vein, and in here I have the secondary capillary net. And this TRH will be released in here and travels through the blood, through the hypophyseal portal vein, and it will reach in here, and by the simple diffusion it will reach its target cell. What is its target cell? It is a trope, and it is called the thyrotrope. So the TRH, which is a releasing or a stimulatory hormone, will come and bind to its receptor and go to the target cell, which is the thyrotrope itself. And in here, it will then release the TSH. TSH is another hormone. It is a thyroid stimulating hormone. It will come into the blood, and by the blood current, it will come and reach to the thyroid gland. So these are the cells of the thyroid gland. They are epithelial cuboidal shape, and as soon as the TSH, which is the thyroid stimulating hormone, will connect to its receptor in here, some processes in the cell will happen, and finally the T4 and T3 will be released into the blood. So what are these processes? TSH is a polypeptide hormone. It will bind to its receptor in here. Exactly near to its receptor, there is a G stimulatory protein which is bind to GDP, and it is inactive. But as soon as this connection and the TSH connects to its receptor, GDP will change to GTP, and then G stimulating protein becomes active. Then it will go to another enzyme, another molecule, which is adenylate cyclase in here, and makes this adenylate cyclase active too. So this is an enzyme, and it is called adenylate cyclase. It will come and have some enzymatic processes. What are they? It will change the ATP to cyclic AMP. And this cyclic AMP, which is derived from this ATP, then we have cyclic AMP. This cyclic AMP will come and activate protein kinase A. And why A? Because it is related to ATP, it is related to adenylate cyclase. For example, we have different kinds of protein kinase. This one is protein kinase A. We have protein kinase C, which is related to calcium, but in here we have protein kinase A. And protein kinase is related to phosphorylation of some proteins. For example, there it will go in this example, which we are talking about thyroid gland and also follicular cells, which are the cuboidal epithelial cells. It will come inside the nucleus, and inside the nucleus it will activate the transcription factor, and this transcription factor in fact will become activated because it is phosphorylated. So this protein kinase is phosphorylating some proteins, some enzymes, and it will make them active. And in here it is activating the transcription factor, which is connected to the DNA. And in here the transcription factor will make the mRNA. mRNA comes out of this nucleus and will go, and by the ribosomes it will make some proteins. These proteins will go inside the rough endoplasmic reticulum, and finally they will travel to Golgi, and finally from Golgi apparatus it will have a vesicle, and inside this vesicle there is a protein, which the synthesizing of this protein had been started because of my TSH binding to its receptor. So finally this vesicle will fuse to this membrane in here and will be released this protein will be released in here without its vesicle. Without the vesicle, the protein will be released. So this protein is called what? It has a name, this protein. The protein name is thyroglobulin colloid. This thyroglobulin colloid does not have these iodide yet. It doesn't have it yet. It hasn't been bounded yet. It is without these iodine. It is pure. It is this, in fact, thyroglobulin is like this. It is pure without these iodide. It means that, so this is my pure thyroglobulin, which has come from here. But it needs that iodide will be binding here. But how the iodide will be brought inside this colloid, this part, this lumen is called colloid, and these are follicular cells. So how the iodide is brought in here? Inside the blood, there is iodide, which is negative, iodide which has a negative part, and it is active and it is low in the blood. And inside this follicular cell, the iodide concentration is high. So what happens? I need to bring this, which is in here low, in here high, inside the follicular cell. How I bring it? By the secondary active transport or co-transport or symport. How? Because in here the sodium is high, in a high concentration, so sodium comes inside the cell, and co-transport or symport, the iodide will come inside this follicular cell. And finally, by another pump, which is name is penderin, it will go inside the colloid part, into this lumen, into this colloid place, which we call it colloid. So the penderin pump, this pump name is penderin, and this iodide will come in here. It is so active, so it must be inactive. So from this negative part, it must have a 0 oxidation number. So another enzyme is in here, which is called the thyroid peroxidase. This thyroid peroxidase does 3 jobs. One of its jobs is iodide oxidation. It means that it will change the iodide to iodine and make it inactive, and then it will bind the iodine to these amino acids in here. What is this amino acid? This amino acid, in fact, is called tyrosine amino acid, and then this thyroid peroxidase will make these iodide, iodine to be bound in here and make DIT and MIT. Now I will explain what is DIT and MIT. So finally, these thyroid peroxidase, which makes this iodide inactive and make I2, this is one of the jobs, and the other job is binding these I2 to these amino acids in here, which are 4, and they are called tyrosine amino acid. So and when 2 iodine is bind to 1 amino acid, it makes diiodotyrosine, because it is 2, so it is called diiodotyrosine. And in here there is another diiodotyrosine, which we call it DIT. In here there is one iodine which is connected in here, so it is monoiodotyrosine. And in here I have another diiodotyrosine. Finally, the third job which this enzyme does, which the name is thyroid peroxidase, so this enzyme does, is this, that diiodotyrosine and diiodotyrosine, they will be bound to each other and make T4. The name of the T4 is thyroxine. Another name of the T4 is thyroxine. And it makes one monoiodotyrosine connected to diiodotyrosine and makes T3 or triiodothyronine. And it makes T3 or 3-iodothyronine. So they are connected. And this enzyme has done its job and it's finished. Now I want to insert and bring this molecule, which is a thyroglobulin colloid, inside the cell. By endocytosis, it will come inside the cell. Still, I don't have T4 and T3 because they are connected to thyroglobulin colloid. But there are lysosomal enzymes and also lysosome in here, which has some enzymes in here. It will come and its membrane will fuse to this vesicle, which has come by endocytosis inside the cell, and it will cut the T4 and T3, and T4 and T3 in another vesicle will be released, and finally this membrane of this vesicle will fuse to the cell membrane, and this will by diffusion go into the blood. But it's not that much easy. Why? Because T4 and T3 are like steroid hormones and they cannot travel easily through the blood. So what happens? The liver will make a carrier for this T4 and T3 and bring it into the blood, and this carrier is called thyroxine binding globulin. So thyroxine binding globulin will come in here and it will carry T4 and also T3 with itself, and they are going and traveling to the target tissues, which the target tissues can be bone, muscles. It has several target tissues and it will have its effect. So what happened? I had TRH, which is a stimulatory hormone. It will be released into the hypophyseal portal vein. Finally, I have TSH, which is made by the thyrotrope. It will come into the blood. It is polypeptide. The receptor is here. Some process is happening here. And finally, I have thyroglobulin inside my colloid. Inside my colloid is released. It doesn't have iodine, so iodine must first of all will be brought in here by co-transport or symport and through the Penderin pump. Then the iodide must be iodine. The iodide must be iodine, and there is another enzyme in here, which is the thyroid peroxidase. It does 3 jobs. One of the jobs is making iodine out of iodide. And the other job it does is attaching and iodinating the thyroglobulin colloid, and then it attaches and connects the iodine to this amino acid tyrosine, and finally it will make this thyroglobulin colloid, and this thyroglobulin colloid must come inside the cell by endocytosis. It needs energy, and in here I have lysosome. It has enzymes. It will cut T4, T3 into the vesicle, and finally the vesicle membrane and also plasma membrane will fuse, and T4, T3 will come in here. There are steroid. They are like steroid hormones. They are not steroid. They are like steroid hormones. So they need thyroxine binding globulin, which is made by the liver. It will attach and carry them to the target cells. So all in all, everything about the thyroid gland synthesis, about T4 and T3, I tried to cover every note, and thank you for watching this video. See you in the next video.